right, so we finished up photosynthesis Monday. Today we're going to get into cell communication. We're going to at least try to get through section 1, which is really kind of a big chunk of it. And then we'll start section 2. I'm excited if we're going to do section 2 some tomorrow. Since yesterday you did kind of have it close to 3 days, you did my class and use that to study day. So we'll see, kind of see how I feel. Now we're into cell communication. I'll be honest with you, this is not something that I'm as familiar with as with all the other stuff, and this is 100% just AP Bio. On level stuff does not go over this at all, so there's no like on level correlation, which is why you have the weird assigned reads. All right, so by the end of the section, what we want you to be able to do is know the 4 types of signaling that are found in multicellular organisms. Some of them are going to have weird words. Know the difference between internal receptors and cell surface receptors. What do you think the differences are? What do you think internal receptors are? Inside the cell. What do you think the cell surface receptors are? Outside on the surface. Very good. Also, what's the relationship between ligand structure and its mechanism of action? Mechanism of action is the job it does. Ligand is just a fancy word for saying signal molecule. Okay. Ligand is a signal molecule. All right, so how would you handle communication in the following situations? You would like to tell your friend across the room about a party that is happening tonight, but you don't want to disrupt your teacher or let anyone else in class know about it. How would you get this message to them before the class is over? Yes, ma'am.
Just pass a note or something?
Yeah, pass a note. Yes, sir.
What class we talking? If it's in their class, I ain't saying nothing.
That's fair, right?
Mr.Rowell.
A teacher wants to remind everyone in class that they have a test on Friday. What would be the most efficient way to contact every student?
Tell them class.
Yeah. You're in the yearbook committee and would like to organize a fundraising car wash event. How could you get the word out in the community so there's a good turnout? Right. Your college counselor at a high school and you need to give an application to all seniors who are applying for early admission. How can you notify each of these people?
Email.
Yeah. So here's the thing. This is going to correlate within this cell communication stuff because sometimes you need to tell one cell one thing, right? So that's going to be this example. So there has to be a certain pathway of communication to do that. Other time, you need to tell all cells the same thing at once, right? And so there's going to be a specific pathway to get that done. Now, the way it does it is it can either have targeted ligands, these signaling molecules, or you can have targeted receptors. For example, in the notes passing example, let's say, we'll say the note is a baseball and you're able to throw it across the room, but only because you have baseball gloves. Okay? Does that kind of make sense?
Yeah.
That's kind of the concept. They only have a receptor for that one specific molecule. So how do other types of organisms communicate? It's going to depend. Bees, they've got a unique way they dance.
Really?
Yeah.
I did not know that.
You have video mention that, Mr. Rowell?
No. So they dance certain patterns to tell other bees where there are certain resources, whether that's flowers, shelter, things like that. You can also release pheromones in some organisms. Have you ever seen dogs drag their rear ends?
My dog does all the time.
They're releasing scent glands. That's a method of communication as well, right? So it's going to vary depending on the organism in the cell. Some more examples of short and long distance messages in the body. Any thoughts there? What type of message would be general messages sent throughout the body? What type of messages would be more specific? As well as how do cells communicate with each other if they are far apart? Some of the stuff that we're going to talk about. Yes, sir?
Well, what about when you get like a fever? Is that like a message?
Yes.
Your body?
Yes. Because essentially what you're doing is I don't know exactly what cells of the body are integrated into it, but I'll imagine it's quite a few. Because the goal of the fever is to raise your internal temperature. One of the best mechanisms to do that is with cellular respiration because it gives off heat. The reason they do it is to basically whatever you're fighting, you want it to be at a temperature that isn't optimal for that bacteria to grow. Like say if that bacteria grows really well at 98.6 degrees, well, let's raise it to 101. It's not going to kill it, but it's going to slow it down a little bit. Okay? Does that kind of make sense?
Yeah.
I would say that would be one that's not specific. It's just everywhere. Yes, ma'am.
What about like when you're having to throw inputting? Is that part of it? Or is that
Yeah, you could say it was. That might be more neuron-based, which neurons do cell-to-cell communication because neurons are cells, but yes. Especially because even, great point, like if you touch your arm and you feel it and brain feels it, that's cell-to-cell communication too, between neurons. Okay, that's kind of what we're going to talk about today. But in general, this is kind of how it works. You've got a ligand, which is a signal molecule. Depending on what type of communication, you might have a receptor, The ligand will bind to the receptor, which will cause a cascade of changes and get the response in the cell that you're looking for. Okay? And essentially, once you're in here, it's chemical messages. Essentially, pretty much everything within the body will be chemical messages in some way, shape, or form. So it's industries and molecules moving from one spot to another, sending a message, and it could be just like a chain reaction time. Have you ever seen those Newton cradles where the ball comes here, hits this one, and then the other one goes flying? Kind of the same thing happens in the body, except with these certain chemicals. This ball over here eventually moves this one here, but it has to go through intermediate steps to get that done.
Isn't that kinetic energy?
Yeah.
Don't you have one?
Yes. So there's 4 categories of signaling. There's paracrine, which is moved by diffusion through the extracellular matrix. Examples include synaptic signals and neurotransmitters. So this is just diffusion, going from area of concentration of high concentration to low concentration. So this is how, when we talk about neurotransmitters, this is that transmission of information between neurons, like your brain to the rest of your body, etc. Endocrine, this is going to be hormones. Signals from distant cells typically produce a slower response with a long-lasting effect. These are things like hormones. For example, adrenaline. If your body releases adrenaline, it goes to every cell in your body. And then with relation to how fast other things happen in your body, it's going to be there for a little bit, you know. Because this paracrine signaling, the gap between 2 neurons, think about it like 1/1,000,000th of a millimeter. That's about the distance those things are traveling. Autocrine. Auto means self. So this is cell signaling that can bind to the ligand that is released, such as the signal and target cell can be the same or similar to each other. For example, there's something called cell death signaling, apoptosis. That's where if a cell is getting old, it's starting to get degraded, it will throw out a signal to itself and sometimes the cells that are just like it around each other, and it will cause that cell death. But it does it to itself, so it's called autocrine. And then direct signaling and cause gap junctions. If you remember from, I think it was chapter 1, gap junctions are specialized structures in cells, essentially like a hallway. So if you view cells as rooms, say plant cells because they're a little bit more square, gap junctions are basically doors we can walk through between 2 different cells. These are very, very short, very, very quick. They allow small signaling molecules to move in between cells that are connected. All right, so visually this is kind of what it looks like. Autocrine, the cell targets itself. Signaling across gap junctions, you have a gap junction, it's like a hallway or a door between your cells. Paracrine, the cell targets a cell nearby, so they're going to be really, really, really close to your friend next to you that you can pass a note to really quickly. Then you have the endocrine, this is hormones, where basically you'll have hormone producers, like say the thyroid's one of them, like up here, they can send messages throughout your entire body, so it's really, really far away. The message mode of action, how it gets from here to your toes, usually through the bloodstream. Alright, so paracrine signaling example, it's the distance between the presynaptic cell and the postsynaptic cell. This is called the synaptic gap. This is the connection between one neuron and another. The enzymes in the synaptic cleft degrade some types of neurotransmitters to determine the signal. So essentially, this one will release information across this gap. It will diffuse from an area of high concentration to low concentration. You've got neurotransmitters here that will absorb it, and then it will send a message through the rest of that neuron. But how do we keep that signal from keep on being sent? For example, if we want to move our muscle, does it just stay there because that signal keeps on getting sent, or does that signal get degraded and you can relax it? Okay. Right? We do that because we've got enzymes that destroy that neurotransmission. Some people think this is why charter horses happen, because that neurotransmitter's got messed up. It's just a contract signal. Contract.
Cramp in your like right here.
Yep.
Like hurts.
Yeah. Internal receptors. So hydrophobic signaling molecules can diffuse across the membrane. Why?
Hydrophobic signaling, because aren't the hydrophobic in the inside?
Yeah, so most of the plasma membrane is hydrophobic. If we look back to how the plasma membrane is structured.
Oh, Joe just looked at you and turned around. Yeah, he's going down.
He's getting a note. I forgot to do attendance anyway, so.
Stage.
It was funny because all I see is him like walk and he turns around.
Who else is gone today?
Daniel.
Yeah. Anyone else? Alright, cool. Alright, so we've got the phospholipid bilayer that looks like this. This whole area is hydrophobic.
There he is.
And the head's
Okay.
hydrophilic.
Yeah, and these are hydrophilic.
He's here. He's giving blood.
Bang.
He's giving blood.
No, he was skipping class. Let's be real. They said, hey, what time? What time do you want to? And he goes, well, AP Bio starts at 1:40, so let's pick that time.
Actually, I was there right after lunch. Actually,
And
no,
you just not
no,
get
during lunch.
He just not got done.
So, back to the point up here. Because most of this membrane is what? Is it hydrophilic or hydrophobic? Hydrophobic. Small hydrophobic molecules can easier cross it because this is the biggest gap they have to jump. Because if you have a hydrophilic molecule that you try to put through here,
It'll stick.
it'll push it out. You're like, hey, you're not cool, you can't hang with us, get out. So that's why you can have those small hydrophobic molecules pass through the membrane. On those, you don't need an external receptor. You can just have one on the inside right here. So you've got the plasma membrane. The signaling molecule, also called a ligand, will pass through it, attach to the intracellular receptor, which we'll call as a cascade effect, and it will do something else. So these signaling molecules or these intracellular receptors, they can interact with DNA to regulate gene expression. For example, turn on a gene or turn off a gene. You okay? You look all smiley. If you need to eat something, you can eat something.
I don't have anything.
Oh,
It
don't
had
worry.
stuff in there.
Did you not grab the snacks in there?
It had
I
stuff
ate
in there.
during lunch. It's fine.
They have cookies,
Yeah, they have cookies
pretzels,
and-
and
Right, you
drinks
need
in
to get
there.
your blood.
Yeah.
No, you need to eat something. They
Yeah,
take
go get something.
they take 1,700
You
calories
need
in
to
your
get blood
body.
sugar up. That's not a It's a have to, not a if you want.
All right. Paracrine, again, this is one cell right next to another cell. Occurs between cells that are close together. For example, synaptic signals between neurotransmitters. The neurotransmitter or the ligand is released. It gets attached to a neurotransmitter receptor, and then sends a message on down the line. This animation shows how neurotransmitters are affected by cocaine. Cocaine blocks reuptake channels for dopamine. The increased amount of dopamine makes a person feel good. So if you saw the presentation that Officer Goucher put on,
He didn't put cocaine. He didn't say anything about cocaine.
yeah, he's no, same concept, right? So basically what it does is it blocks it. It blocks the reuptake channels. So essentially, it releases dopamine, and instead of that dopamine being reabsorbed, it just stays there. So you're feeling good. Feeling good. Everybody? No?
Don't quit your day job.
Oh well.
That's all I know.
Again, what type of signaling is this?
The pear.
Paracrine. Para means kind of next to.
Pear.
Like,
Pear means pear.
It means next to or outside. Auto means self. If you remember juxtacrine in your reading, juxt means right next to, like juxtaposition. Alright, endocrine signals. These originate from endocrine cells within the endocrine system and send signals to distant cells. These signals are in the form of chemical messages known as hormones. The hormones will be type of ligand. Hormones act only on target cells. So this is going to be the example where, let's say we have the hormone testosterone, right? Only certain cells will have a receptor for that hormone.
See, Mr. Ice up hard.
Yeah, well, if he passes out, he can't do well. I don't want him using that as an excuse not to take the test on Friday.
Dang. What if I like
Don't get it still.
forget to have my bread cookie?
For example, here, this red spot, what is this red thing?
Bloodstream.
Yeah, it's the bloodstream. It's blood vessel. We've got a secreting gland, an endocrine gland, sends off these hormones or ligands. They travel through to the body to the target cell that has the specific receptor to send that message to that cell. Now, it doesn't hit the ones that don't have that receptor. Hormones are extremely important because they help maintain homeostasis through something called a negative feedback loop by returning the body back to its set point. For example, maintaining temperature, blood glucose levels. For example, if we're sick and we release a hormone or a messenger system that tells us, hey, we need to raise our temperature. And so the cells go into overdrive, doing cellular respiration to raise our temperature. Eventually we need to stop because what happens if our temperature gets too high?
We die.
We die, we'll pass out, right?
Brain cells.
Yep. So essentially, like, our body can't function at that temperature. So we've got what's called a negative feedback loop. That is where you return back to the normal condition. Okay. Now everything in our body, all the messaging systems are either going to be negative or positive feedback loops. Negative feedback loops return to the normal condition. Positive feedback loops go and make the condition higher. Now here's the deal. Positive feedback loops are going to be present within negative feedback loops. Okay? Because you can't just have an uncontrolled chemical reaction like, all right, we're going to increase the temperature, we're going to increase the temperature, we're going to increase the temperature. There's got to be some controlling factor in it as well. To maintain your homeostasis, we've got a gland that releases a hormone that lowers, we'll do this one here. So let's say a specific body condition, temperature. We're getting a little bit cold, so we've got a gland that says, hey, we're getting cold, we need to increase the temperature. We increase the temperature. Like, all right, hey, cool, we're back to the right temperature.
Now
But
we're too hot.
we're getting too hot. So it's like, all right, hey, we need to lower that temperature again, right? So this is that negative feedback loop where we're trying to reach this target condition.
So that's why you shiver when you get cold.
Yes, partially. I don't know the exact physiological response from shivering. I don't know how that's like initiated, but in general, yes, that's a negative feedback loop. Because what happens when you get back warm again? Do you keep shivering?
I do. I shiver a lot.
When you get warm enough, you stop shivering. Okay, that's your body's mechanism of being like, all right, yeah, we're back to this normal condition. If I keep shivering, I'm going to get too hot. Oxytocin increases uterine contractions in response to surface stimulation. This is going to be a positive feedback loop. Oxytocin also plays a role in bonding, and climate change is also a positive feedback loop. So, for example, we've got the baby pushes against the cervix, causing stretch, which causes impulses to be sent to the brain. The brain stimulates the pituitary to release oxytocin. Oxytocin causes the muscle linings to contract, which causes the baby to push against the cervix even more. And then it just keeps on going so that it keeps contracting and keeps contracting and keeps contracting. Yes, ma'am?
Is this the oxytocin? Is that only apply here? Or is these are like a general definition, or is that?
Oxytocin does a lot of different things because it also makes you feel good too.
Oh yeah.
It's supposedly I think it's one of the hormones that gets released like when you get hugged or something. I don't know.
Happiness.
But essentially, as you can see, the more oxytocin released, the more these cells contract, the more the baby pushes against the cervix so you keep on. That's why your contractions continually increase and increase until the baby's delivered. Yes, ma'am. Yes, sir. I was thinking about babies being delivered, and I had female brain.
Speaking of hormones, so what's the difference between dopamine and serotonin?
I don't know off the top of my head. If we have time at the end, remind me and we'll look, okay?
That's good.
One of them like relaxes you, one of them makes you feel happy. I don't know exactly. We'll look, okay? Autocrine signal is a self-signaling where the cell will send its own signal to itself, or very similar cells right next to each other. Signaling and target cell are very similar or the same. For example, a cell that is infected with the virus can signal apoptosis. This is cell death. This is an important term you should probably know. In itself, enabling cell. So a single cell receives a weak autocrine signal. This one receives a signal saying, "Hey, I need to undergo apoptosis." And then all the identical cells will receive that signal, and then they will also start to undergo that apoptosis, that programmed cell death.
Crouch up there. Yeah, I'm gonna kill it.
I say.
Yeah. Direct signaling. This is kind of through those gap junctions that are going to look like this, where cells are connected through these gap junctions, and then you can just send those signaling molecules right through. You don't need an external receptor, just kind of an internal receptor. You're going to see this a lot in plants, and the reason you see it a lot in plants is because you've got a cell wall in plants. It kind of inhibits basically the movement of molecules because it's a little bit more of a solid barrier than a cell wall or a cell membrane. So like the cell membranes are fluid, permeable. A cell wall, not so much. So you have to have these special things called plasmodesmata, which allow communication between these cells.
There's too many big words.
Yep. It's a lot. I think next time I do this class, I'm going to have y'all learn root words because it helps.
Or just have them do bio first.
Yeah. That wouldn't help. Or take biology first, is that what you meant? Alright, so again, endocrine signaling, basically you send a message usually from a hormone throughout the entire body to another cell that's a long ways away that has that target receptor. Paracrine is really close to each other, down a concentration gradient. Autocrine is self-signaling, and then the other one is that direct cell signaling where they're connected to each other and send that information over. Alright, so this is kind of a generic signal pathway. You've got your signal or ligand and a receptor. That receptor will basically say, hey, I've attached to the signal molecule, I need to do some work inside the cell, which causes a cascade of reactions, which can change various things within the cell. For example, it can be like, hey, we need to speed up cellular respiration.
Alistair, could you please go to the fax room?
We gotta get blood.
It could be something as a gene regulator. It's like, all right, we're expressing the gene for example, in, you know how certain animals, like the snowshoe hare, can change color during the year? You're like, hey, it's getting color, it's getting colder, we need to change our color from brown to white. So we're going to express that white colored gene. Cytoskeleton protein, this is to alter the cell shape or mobility.
Can you make girls get white hair when you're stressed? Or overstressed? You can get white hair.
To an extent, yeah.
Right
I don't know
here.
the exact.
Right.
I don't know the exact mechanism, but it has something to do with the maturation of something called melanocytes within the hair. And I assume stress releases certain hormones that can accelerate that maturation process. Yes, ma'am.
So you know how when like kids, like when they're little, and then like they grow, like their hair color changes?
Yes.
Because mine was like very blonde.
Yeah, I was blonde too.
Really?
Yes,
Like
I
why
was
does that
blonde.
happen?
I think it has to do with the maturation of the hair follicles.
So is Michael's like blonde.
Because my eyes also get darker too.
What about for like this? What about for like chameleons?
Oh, yeah.
They can like.
I mean, there could be some type of cell signal. I'm not sure if it's going to be gene related, but it would be more along the lines of probably an altered metabolism of some sort. Basically, like, hey, we're going to break down this molecule because it produces this color. But in general, the signaling would still be the same. As
Sorry, with like remember the waves thing where it like reflects?
Yes.
Is theirs like? Do you know anything on that? Like, is it all the same, or can they like shift?
They shift it, right?
Okay.
They shift what pigments they show to absorb or reflect a certain amount of lights. And so essentially it's probably, I'm going to say it's a metabolic pathway. They have to produce some type of pigment or break down another type of pigment that basically reflects or absorbs whatever wavelength of light they're trying to do. Okay. Does that kind of make sense a little bit?
Yeah.
So the graphics line in the short video. Do you want to watch a video?
Yes.
Oh, yeah.
Yeah, let's see if we can get it to work.
Which one?
The action of epinephrine. Epinephrine.
Oh, it's only a minute.
Epinephrine is one of many hormones that is water-soluble, hydrophilic, and therefore unable to cross the hydrophobic plasma membranes of its target cells. Instead, it binds to receptor proteins located in the plasma membrane and does not enter the cell. When epinephrine binds to beta-adrenergic receptors on the liver cell, G proteins on the inner side of the cell membrane are activated. Each G protein is composed of 3 subunits, and the binding of epinephrine to its receptor protein causes one of the G protein subunits to dissociate from the other 2. The G protein subunit, which dissociates from the others, carries a GDP, which is replaced by GTP when the subunit is activated. The activated G protein subunit then diffuses within the plasma membrane until it encounters adenylyl cyclase, a membrane enzyme that is inactive until it interacts with the G protein subunit. When activated by the G protein subunit, adenylyl cyclase catalyzes the formation of CAMP from ATP. The CAMP formed at the inner surface of the membrane diffuses within the cytoplasm, where it binds to and activates protein kinase A, an enzyme that adds phosphate groups to specific cellular proteins. In liver cells, protein kinase A phosphorylates and thereby activates another enzyme called phosphorylase, which converts glycogen into glucose 6-phosphate. The glucose 6-phosphate is then converted to glucose. Through this multi-step mechanism, epinephrine causes the liver to secrete glucose into the blood during the fight or flight reaction.
Why would you want to have glucose in your blood during the fight or flight reaction?
Because it gives you more
It
energy,
gives
so
you
you can run
power
faster.
to run.
Because it gives you more energy. All right.
Neurotransmitter receptors do not
Shame
have
on videos.
That's right, you know what's funny?
Instead,
You could have pulled that up and your channel came up.
Yeah, no, I don't know about that. Yes, ma'am.
Oh, question. So,
Burger King, Montana, Haiti, and Tanner had to come to the office for you.
so you know how octopus change color like they camouflage?
Yes.
How does that happen?
I think it's the same way chameleons do. They're able to do certain metabolism within the individual cells. They call it the reproduction or the removal of a pigment. They either produce color change or stop color change. That's my best guess. Okay. All right, so you should be able to know all the steps of epinephrine. Okay. What time is it? 2:00?
Like actually.
Yeah, absolutely. Definitely. Definitely.
Why are you taking photos?
Bro.
All right. No, you don't. You don't have to know all the steps, but you do need to know. You know, it starts with a hormone, and then you have all of these step-by-step reactions that happen within the cell, right? So,
Okay, I was actually typing.
no, you don't need to know that. I was being facetious, okay? All right, so receptors. There's two types we're going to talk about. Cell surface receptors, which there are various subtypes, and there's ligand-gated ion channels. Okay. It says you need to remember the CFTR channel. We'll talk about that in a little bit.
Yay.
All right, cell surface receptors. It can be ion channel linked receptors, G protein linked receptors, or enzyme linked receptors. Get out of my house. As I said, was anybody in here? No, nobody's in 10,000 biology last year.
I was just trying not to make a mess. And facts I work. You're the one making
Yeah,
the
5th hour
mess.
last year.
Oh, I heard about it. You made like a bet with something.
Yeah, we did attention if they had the water bottle and made it the trash can. It was Haley Allen, and then she threw it from over there, and I was like, "See you for next week." All right, then you've got enzyme linked receptors. So, ion channel linked receptors. They receive a signal molecule or a ligand, and they open up to let ions through, right? Which is going to change what? We let ions through a semi-permeable membrane. What changes?
The gradient.
The gradient. Very good. You've got G protein linked receptors. That's kind of the one about epinephrine. They receive a signal molecule. They activate this G protein, which causes them to activate other proteins to get stuff done within the cell. And the example there, it was the release of glucose into the blood. Then you've got enzyme linked receptors, where your signal molecule comes in, turns into a dimer, and then you have these two proteins attached to each other. Enzymatically attached.
Yes.
All right. Cell surface receptors, example, ion channel linked receptor.
Oh, Cameron!
So you got gated ion channels from a pore. You want to let him in?
Yeah.
No.
Yeah.
Okay. You can talk, but I'm going to keep going.
Camera.
Okay.
Hey, Mr.Rowell.
Yes, sir.
Whenever this period ends, and you're good to go.
Okay. Are they staying this late?
I think so, yeah. Because there's still a lot more people who are waiting.
Cameron,
Hey, that'll
you
work.
see
I like it.
AP
Thank you,
bio?
sir.
Oh, there's AP Bio?
Yeah.
Yeah. Hey, first year, it's not going great.
Oh.
All right. You've got gated ion channels, which form a pore through the plasma membrane that opens when a signaling molecule binds, allowing these ions to go through, which is going to change what, Mr. Pence? The concentration gradient, which is going to change things within the cell. This is an example of the G-link protein. Again, you have that hormone or ligand bind outside. It activates a G protein, which goes on to activate other enzymes and proteins within the cell, causing that cascade change. Did you say something?
I literally just learned this in my biology.
Good deal. See?
He's in college level biology.
That's what AP biology is, Mr. Pence. that makes me feel better, because his biology is one semester.
Yeah. What? You guys taking this all year?
Yeah.
Ha. Well, I gotta learn all this in one semester.
Yeah. So keep that in mind. All the stuff that we're learning this year, he has to learn in one semester. How many days a week is your class?
3.
3. In 3 days a week.
Yeah, but the cool thing is I get a lab every Monday.
Yeah.
We're supposed to to do our lab like every Wednesday.
Yeah, that didn't happen.
Yeah.
Maybe on the next day for biology class.
I got to make bread. It was great.
That's it?
Yeah.
Alright.
Yeah, that was about to tell you. That was like respiration.
Pretty sure the only thing we've done in lab here was Garrett wrote 2 beakers in 2 seconds on the first day.
Hey, don't worry, someone broke a beaker first week of ours.
Hey, alright.
You're not alone.
Okay, it looks so different than like.
Why are your eyes like?
I don't know. I think it's the hair. It just looks so weird.
Definite downgrade.
Yeah, definitely. You definitely downgrade from whatever.
Yeah. This is just another visual showing that G protein coupled receptors, where you see a receptor or a chemical signal bind on the outside. It sends signals into the inside of the cells, which causes further signal changes. For example, we talked about that epinephrine causing the glycogen to be released into the cell. Okay. Just another visual there as well.
It's so simple.
I stole this from another PowerPoint presentation. I
Plagiarism.
absolutely. They posted online for everybody to share.
You already talked about integral proteins and.
Yes. Anybody want to tell them about integral and peripheral proteins?
Yeah.
Nah, just on the outside
They're loosely attached to the outside.
and inside. Integrals go where?
They're like crisscross.
They all look
through.
very.
What are the peripherals attached by?
I don't remember. It's like it's not. They're like it's like a cord or type chain or something that connects them to the outside, so they're loosely attached.
Isn't
Oh,
that
yeah.
that was like linked by anchored by lipids?
Yeah.
Usually. All right.
No.
Please do.
I was about to say, like, somebody said no. Anybody want to watch it?
We should just keep going.
Just have me teach it.
Hello. Today in this video, I'm going to discuss about G protein coupled receptors, their
This is
structure,
the voice of like
and the negative
on
impact.
YouTube.
Protein coupled receptors, or GPCRs, are transmembrane proteins made up of a single polypeptide chain and span 7 times through the plasma membrane. Each receptor has an N-terminal extracellular domain and a C-terminal intracellular domain. Ligands such as peptide hormones, which cannot pass through the cell membrane, bind to the extracellular domain and trigger intracellular signaling cascades, which mediate the functions of that particular ligand. G proteins are a group of membrane-resident proteins which recognize the activated GPCRs and relay their messages in order to elicit the cellular effects of the ligand. G protein consists of 3 subunits: alpha, beta, and gamma. The gamma subunit is anchored to the plasma membrane via a fatty acid chain. In the resting state, G protein exists as the alpha-beta-gamma trimer, which may or may not be pre-coupled to the receptor. And in the resting state, GDP is bound to the alpha subunit of the G protein. When an agonist molecule binds with the receptor, a conformational change occurs. This causes interaction of the receptor with the G protein. This reaction occurs within around 50 milliseconds. GDP dissociates from the alpha subunit that GTP binds. This causes the activation of G protein and release of the alpha GTP fragment and beta-gamma dimer from each other. Alpha GTP and beta-gamma dimer are the active forms of the G protein. Alpha GTP combines with effector enzymes like adenyl cyclase and phospholipase C and activate them. Beta-gamma subunit mainly binds to ion channels or kinases and activate them. There are 4 types of pharmacologically important G proteins have been identified. They are G-stimulatory, G-inhibitory, G-node, and GQ proteins. Now let's discuss a bit about the adenyl cyclase pathway. Adenyl cyclase is a membrane-bound enzyme which is activated by G-stimulatory proteins and inhibited by G-inhibitory proteins. When activated, it catalyzes the conversion of ATP into cyclic AMP. Cyclic AMP activates CAMP-dependent protein kinases, and these kinases cause phosphorylation of certain enzymes according to the agonist molecule which bound to the receptor. In the phospholipase C pathway, when the agonist binds with the receptor, it activates a GQ protein.
Alright, I'm not going to make you watch the rest of it. Listen, I'm going to be honest, it's a little bit more in depth than what I feel you need to know. Big picture, what do G proteins do?
Activate and inhibit.
Yeah, they can activate and inhibit based on a response from what? What's the first step? Something has to come in there and bind to
A receptor.
an extracellular receptor, which then in turn G protein can recognize and then cause further internal reactions to either make stuff happen faster, to slow stuff down, activate, inhibit, or do other things within the cell, right? It's a big picture of what I want you to focus on. Enzyme link receptors. We've got this tyrosine kinase. It's an enzyme link receptor with a single transmembrane region. So we've got two of those there. Binding of a signal molecule to an extracellular domain causes the receptor to dimerize. Di means what? Two. Okay. Tyrosine residues on the intracellular domain are then autophosphorylated. What's autophosphorylation? Or what's phosphorylation? You add a phosphate group. So auto means automatically or self, depending on the condition. Triggering a downstream cellular response by the phosphatase that removes the phosphates from the phosphotyrosine residues. So essentially, I'm going to talk about it being dimerized. It kind of pairs up with another thing, and then it sends those signals on the inside of the cell. Okay. All right, signaling molecules. Small hydrophobic ones. Can these pass through the membrane on their own?
No.
Small hydrophobic?
Yeah.
Yes, usually they can. So steroid hormones have similar chemical structures to their precursor cholesterol. What is cholesterol used in? The no, cholesterol's not used in the cholesterol. Where do we see that? Do you know, or are you just guessing?
I swear you said something.
Okay, it's in the plasma membrane. What does it do?
Oh wait, doesn't that have to do with like letting stuff through fluidity and like
Fluidity, right? So cholesterol only used in animals, and it helps with membrane fluidity under certain conditions and temperatures. Basically, it prevents it from changing too much under temperature changes. Okay. So these hormones that have cholesterol as a precursor change, they can directly diffuse across the plasma membrane because they're going to be relatively small and hydrophobic. There also are some water-soluble ligands. If they're water-soluble, those hydrophobic or hydrophilic? Hydrophilic, very good. These can't pass the plasma membrane as well, so they're going to bind to cell surface receptors. Where are cell surface receptors at? On the surface of the cell, and certain gas ligands as well. Okay. Any questions on this? All right, so these are just kind of examples of cholesterol derivatives. You've got cholesterol, which is a parent molecule, testosterone, and estradiol. Again, since these look like cholesterol, and cholesterol is in that plasma membrane, they can pass through it relatively easily, so they don't have to have specific receptor proteins to do that.
I don't think on my course might be this almost kind of stuff, but we covered the kind of the basic stuff.
Yeah, y'all don't have as much time either, and also like they expect you to do a lot more stuff on your own.
Yeah, we just got through cellular respiration, and we're getting into cell cycle.
Okay. Weird.
Already did
We did cell respiration. We haven't gotten to cell cycle yet. Once we get through this, then we're going to get into mitosis. Not my fingers, but mitosis.
Are you trying to?
Are you still trying
to get
All right, let's do this practice question. Unlike living cells, many viruses do not have a plasma membrane or any of the structures necessary. Say what?
Push in your chair, Cameron.
No, he's fine.
I'm an adult.
No, he gets to do it once.
That's not fair.
Life's not. You get used to it.
what if I'm a
That's
senior
what
in
we're
class and I take speed?
Sure, you can get out of class and never come back. All right. Also, he's never had it as a teacher, so he's right. Unlike living cells, many viruses do not have a plasma membrane or any of the structures necessary to sustain life. Some viruses are simply composed of an inert protein shell containing DNA or RNA. To reproduce, the viruses must invade a living cell, which serves as a host, and then take over the host cellular apparatus. But how does the virus recognize its host? Viruses often bind to cell surface receptors on the host cell. For example, the virus that causes influenza, the flu, binds to receptors on membranes of cells of the respiratory system. That's why you have trouble breathing when you have the flu. It's because that's where the virus binds, right?
It's only after I got the shot.
Chemical differences in the cell surface receptors among hosts mean that a virus that infects a specific species, for example humans, do not infect other species, for example chickens. Usually. However, there can be mutations across that. That is how we got the old coronavirus. Right. And chicken
I thought
pox.
those bats.
Yeah, it's species, not chickens. We also got the bird flu there was a while back. However, viruses have very small amounts of DNA or RNA compared to humans, and as a result, viral reproduction can occur rapidly. Viral reproduction invariably produces errors that can lead to changes in newly produced viruses. These changes mean that the viral proteins that interact Cell surface receptors may evolve in such a way that they can bind with. I lost my spot. The receptors that can bind to receptors in a new host. Such changes happen randomly quite often in the reproductive cycle of viruses, but changes only matter for viruses with new binding properties comes into contact with a suitable host. In the case of influenza, this situation can occur in settings where animals and people are in close contact, such as poultry and swine farms. Once a virus jumps to a new host, it can spread quickly. Scientists watch newly appearing viruses called emerging viruses closely in the hope that such monitoring can reduce the likelihood of global viral pandemics. Just to let you know, this question was written. This book was made.
Question?
Yeah, this was written before coronavirus. Like this is our book's not that new yet. What requirements must be met for a new virus to emerge and spread? The virus must infect at least 2 different animals before infecting humans. The virus must come into contact with a new host, and mutations will occur, which allow the virus to bind to that host. A mutation must occur in the host, allowing the virus to bind to the host, or the mutation must occur in the virus, allowing the virus to infect a new host, and the virus must come into contact with this host. All right, throw up your numbers.
There's multiple.
Okay, I can't see your number, Mr. Ray. All right, cool. Okay. Correct answer is 4. Mutation must occur in the virus, allowing the virus to infect a new host, and the virus must come into contact with its host, right?
Oh my gosh.
So,
Okay, I was gonna say that's another one.
no, this is it, right? Where do we see that up here? We talked about producing errors that can lead to new produced viruses. These changes mean that proteins can interact with surface receptors and evolve such a way that combine receptors in a new host. They happen randomly, but the changes only matter if the virus with a new binding property comes in contact with a suitable host. Okay. HR2 is a receptor tyrosine kinase. In 20% of human breast cancer cases, HER2 is permanently activated, resulting in unregulated cell division. That's what we call cancer. Lapatinib, a drug used to treat breast cancer, inhibits HER2 receptor tyrosine kinase autophosphorylation, the process by which the receptor adds phosphate into itself. This reduces tumor growth in patients by 50%. Besides autophosphorylation, predict which of the following steps would be inhibited by lapatinib and could be documented in its usage. Would it be the 1, dimerization and the downstream cellular response, 2, phosphate activity, dimerization, and the downstream cellular response, 3, signaling molecule binding, dimerization, and downstream cellular response, or the downstream cellular response only? So what's it do? Let me see.
Yeah, I don't even know what dimerization is.
6 2 proteins together.
Okay, yeah.
Notes. Okay.
Just sat down.
I'll give you 4.
What?
Yeah.
I thought it'd be
Yeah.
talking about the
Well, so it doesn't talk about it inhibiting dimerization. It just keeps it from phosphorylating. And phosphorylation is what causes all these downstream cellular responses. Okay, so it's like the stopgap in the thing. All right, that's it.
Perfect timing.
Thank you very much, ladies and gentlemen. Have a good day.
Mr.Rowell, you think I'll get in trouble if I take this Band-Aid off?
Oh, I saw your email. Study one. I checked the syllabus for the extra credit opportunities. Okay, because there's 2 weekly worksheets, and then there are scientific reads, and then also help you do some of my notes.
Where do I look at this syllabus?
You don't have it.
Okay, then yeah. I was like,
So, yes, you're saying that because I put in the chapter review questions from last week, aren't you? Or from 2 weeks ago that everyone's like, yeah, don't put those in yet, and then I finally put them in. Yeah. Okay.
I gotta do this. Preview questions.